Hey everyone, welcome to Taking the Pulse, a healthcare and life sciences video podcast. I am Heather Hoops Matthews here in the studio today with Maynard Nexsen healthcare attorney Matthew Roberts. Matthew, it's good to be back in the studio.
Yes, back in the studio.
We've been on the road for a bit.
We have.
Good to be back. Joining us today is Dr. Leo Ferreira. He is an assistant professor at the Medical University of South Carolina. Leo is also co-founder of Torpedo Bio, which is focusing on solving the unmet need of efficacious and safe cellular immunotherapy for solid tumors, particularly pancreatic cancer. We recently had the pleasure of getting to know Leo and the Torpedo Bio team during the inaugural SC Bio Drive, which is an accelerator for emerging life sciences companies. And Matthew, I know you and the Maynard Nexsen team served as mentors.
That's correct, yeah. We got to meet with Leo and the other folks there. Very impressive group.
Yes, well, we're grateful that, Leo, you took the time for joining us today. Thank you so much. Start us off by telling us a little bit about the company and your role.
Yes, so first of all, thank you for the invitation to be here today. It's really an exciting time for us. So the company really focuses on a largely unmet need, which is to have safe and efficacious cell-based therapies for solid tumors. So you might have heard a lot about immunotherapy. There's antibodies, for example, that will block, how T cells go to sleep. And so if all your T cells are now awake, they're more likely to kill the cancer cells. Now that has severe toxicities because if you wake up all your T cells at once, a lot of unwanted side effects happen, in addition to sometimes a tumor remission. So then people have switched to, okay, so now we can take the T cells out of the body of the patient, right, the T lymphocytes, arm them with a new gene called a chimeric antigen receptor, or CAR for short, and now that CAR actually sees tumor cells. So you have a specific interaction between the CAR T cell and a cancer cell. So then when you make a lot of those cells in a laboratory, you can expand them 100, even 1,000 times, and inject them back into the patient, they now go seek and destroy the cancer cells in the patient's body. Now that has worked really, really well for liquid tumors, leukemias, lymphomas. You have up to 90% remission rate in people that receive CD19 or BCMA CAR T cell therapy. MUSC here, where I'm located, we have a clean cell therapy facility, and people have undergone these trials, and again, you see excellent outcomes in remission, and also the safety has gone up a long way since the beginning. So not so much more or much reduced cytokine release syndrome, neurotoxicity, all these things. So that part has been ironed out, still ongoing, of course, but there are now 7 different FDA-approved CAR T-cell therapies for leukemia and lymphoma. Now, where the technology has not gotten yet is solid tumors, which unfortunately to us are also the deadliest and most common type. Roughly 90% of cancer cases are solid tumors, and that's lung cancer, pancreatic cancer, breast cancer, and others. And so that's where we're entering the field, is that why is it that solid tumors, which again are 9 out of 10 cancer cases, and they're the deadliest types, lung cancer being actually the number 1, why have they not benefited as much from this engineered T-cell therapy? And it really comes down to the fact that solid tumors have a unique biology. They are very good immunologists themselves, if you will. They learned all the tricks to really evade the immune surveillance and to protect themselves from immune attack. And so they build this fortress around them, what we in the field call the tumor microenvironment. And this tumor microenvironment has physical, chemical, and biological barriers for T cells and CAR T cells to ever even go in. And if you do make it to the solid tumor, they become exhausted and they just die once they're there. So we're designing a new immune engineered cell type that can infiltrate and eradicate solid tumors. And so that's where Torpedo Bio comes in, is that if you have an engineered immune cell therapy that can target this largely unmet need to target solid tumors.
So Leo, you've just mentioned CAR T therapy and how it's been successful in other cancer types of treatment. So without giving away any intellectual property, can you talk about how your technology is different? And you referenced it's one way it's different is it focuses on the solid tumors. But how is it different? How does it achieve things that prior attempts have failed? How is yours different?
Yeah, so what we did to address this unique problem is we kind of took a step back and thought, okay, how is the problem starting, to begin with? And so the problem is because, again, solid tumors, they surround themselves of inhibitory immune cell types that prevent T cells from ever taking off. They also create physical barriers. There's a lot of collagen deposition and also erratic blood vessels that go into solid tumors. So there's all these mechanisms that solid tumors have come up with to resist the immune system because, all of us every day probably we have some cells that try to become cancerous. But most of us, fortunately, don't have cancer. Why? Because they get caught so early that those tumors don't stand a chance. Those tiny, tiny tumors, right? But ones that do, that they grow, and then it becomes a gigantic problem, right? And so we're trying to address, like, what is that allows those tumors not only to start, but actually to take hold. And so it's that the immune system was not able to eliminate that early enough. So then when it comes to the clinic, you're going to have a detectable tumor mass. So now how can we infiltrate that tumor mass? And so the conventional T cells, they don't go in because there's multiple reasons. One is to do with, if you will, the zip codes that they have. And so we call these, there's these chemicals called chemokines. Chemo means chemistry and kine means movement in Greek. And so there's these molecules that go in your blood and they kind of tell immune cells where to go. So different chemokine gradients can guide, can direct immune cells to different places. So conventional T cells, looking at their gene expression, at their portfolio of proteins that they have, lack most of the chemokine receptors, the homing receptors that are needed to infiltrate solid tumors. So that's one problem that we're addressing. Another one is the survival once they get there. So the few that do make it to tumors, and we know this is possible, this exists, there's a therapy called TIL, tumor infiltrating lymphocyte. And so if you do a surgery and take a piece of tumor, you do find some few T cells in there. Now, whether they're functional or not is a different question. They are mostly dysfunctional. So that's just problem number two, is that okay, maybe a few of them make there, but then they don't work once they get there. So this new cell type it actually survives and thrives in this otherwise hostile tumor microenvironment. And then the third step, of course, is the target. Where is the target? So our laboratory is not only about the new immune cells, but also the new receptors for them. So we have a whole pipeline to also go after new targets, new surface proteins that might be advantageous to target in solid tumors. And so by having a good cancer target and having cells that get to the tumor and survive and work once they're there, that's really, and then one fourth thing, if you will, is more mechanistic, but the way it works is it's not just engineered immune cells themselves that do the killing, they also call reinforcements. This is what we call bystander suppression. So the bystanders, they now call into action. So this bystander activation, in this case, so suppression would be what the tumor already does, right? So bystander activation, you're stimulating the other immune cells to come in. Because you hear a lot about cold tumors, cold meaning that they don't have an inflammatory response taking place, so they just thrive and they grow and grow. And so we're turning those cold, hard to treat tumors into hot, easier to treat tumors. And so that's actually a big part of the technology, is to really remodel, rewire this
Right.
fortress, this your microenvironment to make it amenable for immune cells to go in and penetrate and destroy the tumor.
Leo, you mentioned your research, and your team bases your research out of MUSC. How did you get connected there?
Yeah, so it's been a long path. I'm originally from Portugal. I did my college there, actually, my bachelor's in biochemistry. And then when I was 20 years old, my parents were tired of me, so they sent me to the United States. And I did my PhD at Harvard in Boston, or I guess in Cambridge more precisely. I had to go to Boston like maybe once a month. Uh, so I was there for 5 years. So that's why I started learning immunology. So my PhD was all immunology and genome editing and T cells. So that's really where it started. And then from there, I had a postdoctoral fellowship at UCSF, University of California, San Francisco, so across the country. And so there we learned more about the immune therapy. And actually, my postdoctoral advisor, former postdoctoral advisor, is now a CEO of a biotech company himself. Um, and so then from there, I learned, so I had the immunology, I had the therapy, and the cancer part. And then my laboratory, my own laboratory now at MUSC in Charleston, since 2021, we started working on this. And so we started seeing these observations early on. Already in 2021, we started seeing them and we prepared that patent application. I'm actually the only inventor in it because back then my lab was just me and then a technician also getting started. And so that's really where we started. So I think in the end, long story short, a lot of discoveries are made out of serendipity, investigating something, and sometimes you find something else. Like I was working on this immune cell type, I was like, oh, there's these properties that could be interesting. And then you kind of connect the dots as, okay, let's try in tumors, solid tumors, we see this because of that. And so you're kind of connecting all this together. And so it's been a very fun and high speed, also stressful sometimes journey. But everything so far has really been lining up to kind of have this novel, unique, engineered immune cell therapy for solid tumors. But I myself, I've been studying immunology for, I think, 12 years now.
So you started your lab at MUSC during COVID. That must have been an interesting process in and of itself.
Yeah.
So as a startup entity, which Torpedo Bio is, what would you say is the biggest challenge that you face as a startup life sciences company? Is it achieving the technological or clinical goals that you started out to achieve, or is it raising enough money, or is it getting regulatory approval? Which of those are the biggest challenge to date?
Yeah, that's a hard question in the sense that I think the science is hard, and raising the money to do it is also not easy. And then, of course, the truth in the end is in the real world, so in the clinic. And so that's what I will say, though, is that we've been having tremendous support on all fronts. So in the Charleston, South Carolina ecosystem, we've received support to start the company, to apply for our first funding, the legal infrastructure, the marketing, and also the clinical part. So MUSC, as I mentioned earlier, it has a clean cell therapy facility which can basically fabricate these cells from patients. So the patient goes in, the blood gets collected, you have the cells, you genetically modify those cells, expand them, and then put them back in and follow the patient. And so MUSC has that infrastructure for at least for a phase 1 proof of concept study to be done in the clinic. And so that's really the next step. So then the challenge right now is we have this solid set of data in the laboratory, right? I have a research laboratory that have these cancer models. We try to be as rigorous as possible. So we use both human cells and also mouse cells. They have pros and cons. The human cells, they're from us, so we care most about them. But then there's only so much you can do with them in terms of readouts, because the immunodeficient mice that we use to put these cells in, they don't fully reproduce the complex tumor microenvironment immune suppressive mechanisms. So then we also use mouse cells where the mice, the immunocompetent mice, so they reproduce the microenvironment in all its, colorful, vast, constellation of immune cells. And of course, mice are not humans. And so we kind of always run both in parallel so that we're really convinced that what we're seeing is really applicable and translatable. So having that, they're now completing over next months to come, then to go into the clinic is the next step. And so that really takes two things. One is infrastructure, and the other one is the funding to do it. So to make, as a term of comparison or as a number, it costs roughly $50,000 to generate a dose of CAR T cells for a patient. And so you could imagine you need roughly $500,000 to do 10 of those, to have some numbers to start seeing something. And so that's, I would see, probably is the biggest challenge. That some people call it the value of death, which you find something really cool in the lab and you start the company and you have it going. And then from there, how do you put these cells into a patient and see how they behave? So I would say that's probably been true for the past 20 years. That's still the biggest challenge.
Right. I can see that.
Yeah.
So that would be considered a phase 1? Phase 1
Correct.
That you're in? And you think it'll last several months?
So the phase 1 trials, it really depends how you design it. I mean, so sometimes the phase 1 trial is only one type of cancer, sometimes multiple kinds, because phase 1 is mostly focused on safety. And so there's some good, work to build upon. We're not the first to inject immune cells into patients. We're not the first to inject immune cells into cancer patients. And so again, the infrastructure, a lot of it is there to support us and to give us kind of a push to get started. But that said, of course, every new product has to go through the similar hoops, right? You have to talk to the FDA, they have to get approved, and then we have all these safety and quality controls in place, which again is a very different world in terms of time that it takes and cost that it requires between the research in the laboratory and the startup company, and then the clinical in the clean cell therapy facility with xeno-free reagents, and then following the patients for, for several months to several years, right?
Yeah, and you're getting help from MUSC as from the clinical side as you go through these, this phase one and hopefully the future phases. Is that because you're doing it with their patients, I guess?
Yes, and one of our co-founders, Dr. John Rango, is a physician scientist at MUSC. Actually, his office is next to mine, and his laboratory is also next to mine.
Okay.
And he sees patients, and is actually the director of the Hollings Cancer Center, access to clinical trials. And so, really, a big focus also of the university as a whole is to really bring discoveries to patients. And so, Torpedo Bio is in this exciting up and coming microenvironment of, we have these exciting findings in the laboratory. Let's bring them to patients. And as you mentioned, right, we have the patients, we have the hospital, we have the facility to manufacture the cells is actually expanding this year of 2025. So they're making more space basically to make more cell therapies. And so, yeah, so at this stage, everything is being built as we speak.
Good, good. There's been a lot of talk in the news, Leo, in the last several weeks about reduction in funding, possible reduction in funding at the federal level of clinical research, primarily through the NIH, but through other agencies. What I think most people don't realize is how big of a player the federal government is in promoting clinical research. But if there is, and medical research, if there is a reduction in funding, what impact could that have on the future of the development of life sciences startups in the United States?
I think the impact could be devastating because in the end, people go where the money is. And so the United States has been and still is a powerhouse for science. I mean, think how many people, I guess I'm an example, come from all over
Yes,
the world.
you are an example.
Right, start their careers here. Why is that? Well, because we have the best research universities, we have the most amount of funding, right? We have the NIH, we have the DoD, we have other institutions as well that provides funding. And if you look at the drugs being developed, I always like to think that a lot of drugs that are distributed all over the world were developed here in the United States. And so sometimes, we have different healthcare costs across the world. And I feel like in the US is higher. Sometimes I wonder because, well, if we have to invent the drug here and test it and develop it and then finalize it, then implementing it has got to be 10 times cheaper than coming up with it to start with.
Right.
And so I think so the federal funding comes to play because when you start, you don't know what's going to be. I mean, the people, think about the Nobel laureates from the United States, the CRISPR Nobel Prize, the Cancer Immunotherapy Nobel Prize. I mean, these people, when they started, Jim Allison for the immunotherapy to have to keep it cancer specific, when he was in Texas an undergraduate, people didn't even know what T cells were very much. And so imagine how many decades of federal funding had to go into understanding what are these cells that are around your blood. They're round, they look kind of boring, probably don't do anything. Turns out they can kill cancer cells, so they actually do a lot. So much that I think 90% of immunologists work on T cells. And so to have that transformation of knowledge, you need a sustained amount of, funding. And it has to be, quantity also plays a role here because in the end, out of 10 ideas, 9 will fail. And
Right.
so what you can have is make it come down to a level where it becomes prohibitive to start something new. And then people will go elsewhere. Now you see, I subscribe to Science and Nature, which are the biggest scientific journals. You now have papers in these journals that have zero authors from Europe or United States. And so that means that the transfer of knowledge has slowly but surely been, going on year after year. And so what I would hate to see is for an ecosystem to be destroyed because, it becomes kind of a cooperative process.
Yes.
Like if the funding gets cut in half, let's say, that does not mean that half the discoveries are made. It means that a tenth of the discoveries are made, a 20th of the discoveries are made, because all the infrastructure was, probably already struggling for funding as it is. if it not just disappears, it brings down, it's kind of a
The
trickle
overall
down effect.
chances, yeah.
Yeah, exactly. So a lot of people won't even like kind of cut in the knob. And so also very quickly, it all seems to be affecting PhD student application and acceptances. And that's pretty terrible, right? Because these are the new generation.
It
So
is
if you
terrible.
cut, like it's like when you overfish salmon, there's no salmon left. And so likewise, if you don't let those people start their PhD, they're not going to be successful in Nobel Prize and famous and billion-dollar biotech companies 20 years from now
Yeah.
because they never got the chance to start their PhD.
These therapies and these drugs don't create themselves. There is a very lengthy and frankly expensive process that leads to that.
Well, Leo, we're glad that you're here in South Carolina doing what you're doing. We wish you and the Torpedo Bio team the best with your phase 1. When you have a successful phase 1 conclusion and you're at the next stage, I hope you'll check back in with us. Let us know, and we'll have you back on and give us an update. Sound okay?
Of course, that would be terrific. Thank you.
Okay, great. Well, thank you again for joining us. For those of you who listened today, we hope you learned a little bit more about what's on the horizon with dealing, for sure, with solid tumors, but even more so. And Matthew and I and the whole team look forward to seeing you next time right here on Taking the Pulse, a healthcare and life sciences video podcast.